In this session, we are going to discuss about the biosynthesis of urea, how the urea synthesis happens in our body. But for that, first we need to understand that why we are making urea in our body. So the basic concept is something like this. For example, whatever proteins we are having, if we metabolize them, we are going to cut the proteins, we are going to generate the amino acid. If we cut the amino acid further, one of the product that we are going to generate is ammonia. This ammonia is toxic substance for our body and we want to throw it out. But the problem is this ammonia, because it is toxic, we can't directly send it to the kidney via the blood, because that in that way it is going to spread all over the body. So first we need to convert into a non-toxic form. So this ammonia will be converted into urea. Urea is the non-toxic form of ammonia. And this conversion occurs in the liver. Then this urea, because it's non-toxic, so this urea can easily go all the way in the blood, and via blood this urea molecule will reach in the kidney and will be excreted out in the urine. So now we have got a basic idea that why we are having the urea synthesis in our body, because we want to convert the toxic form of ammonia into urea, and that can be easily thrown out. But how it is going to happen? So there are basically, this entire process is divided into 4 steps. First, let's understand the basic outline behind this. When it comes to the biosynthesis of urea, to understand the core concept, we will divide the tissues into 2 types. The liver I will refer as hepatic tissue, and all the remaining tissue I will say extrahepatic. Remaining means the muscle, the brain, they are extrahepatic. So what happens is, in the extrahepatic tissue, for example muscle, we have the proteins. These proteins will be destroyed on regular basis. When you are going to cut the protein is made up of chain of amino acid. So when we are going to cut, when we are going to do the catabolism of the protein, we are going to get the different amino acids such as methionine, glycine, valine. We are going to get different amino acids, and then we want to further metabolize them, and one of the product that we are going to generate is ammonia. But what happens is, in the extrahepatic tissue, whenever we want to metabolize the amino acid further, what we do is we convert all the different amino acid into a common type of amino acid, and that common type of amino acid is glutamate. So basically we try to convert all the amino acid into glutamate. All the different amino acid, will try to convert that into glutamate. You will understand the reason why. So whether it is alanine, aspartate, glycine, the different amino acid will try to convert into glutamate. And because you are converting the different amino acid into glutamate, this process is called as transamination. So the first step in the biosynthesis of urea, it is going to be the transamination. And this transamination is going to occur in the extrahepatic tissue. Then this glutamate, it's an amino acid, it can easily come in the blood, it's not toxic. So this glutamate amino acid will come in the blood and it will reach all the way to the liver. Now once the glutamate comes in the liver, then from the glutamate we will try to bring out the ammonia. From the glutamate amino acid, we'll try to bring out the ammonia molecules. And because this process, we are removing the ammonia from glutamate, this is called as deamination, and this occurs in the presence of oxygen. So this process is referred as oxidative deamination. So the step number 2, it is oxidative deamination. So we can see like this. What is the first step? The first step is to convert the different amino acid into glutamate. Then the glutamate will go to the liver, and in the liver, in the presence of oxygen, we will take out the ammonia, that is oxidative deamination. And this is actually what we wanted, because ammonia has a capacity to convert the ammonia into urea. So we have sent the ammonia molecule in a form of glutamate. So you have answered one more question here, that why you have converted all the amino acid into glutamate, because glutamate easily donates the ammonia, and this is what we wanted. The rest of the amino acid, they don't give the ammonia easily. If they can give the ammonia easily, they are happily welcomed in the liver. They can directly go to the liver and they can give the ammonia directly. There are such amino acids are there, I will tell you the name, but most of the amino acid, they don't give their ammonia easily. So first we convert them into glutamate, that is called as transamination, and then the glutamate will go all the way in the liver, and in the liver you are going to take out the ammonia molecule, that is called as oxidative deamination. Now in the extrahepatic tissue, sometimes due to some reaction we can have the free ammonia molecule, maybe in the muscle, maybe in the brain, we have some ammonia molecule, and that has to be brought all the way to liver. because liver has the one that has the capacity to convert the things into the urea. So how we are going to do this? The ammonia molecules from the different extrahepatic tissue, with the help of different transporters, with the help of different transporters, these ammonia molecules will be transported. So the step number 3 will be the transport of the ammonia. From when the ammonia will be transported from the muscle to the liver, it will be with the help of a molecule called as alanine. So alanine is going to be the ammonia transporter for muscles. And from brain, it will be transported with the help of a molecule called as glutamate. So glutamine is the ammonia transporter from the brain. Now with the help of all the transporters, the ammonia molecule has came in the liver. So basically there are 3 ways by which the liver is getting the ammonia. One, the glutamate is giving the ammonia, that is oxidative deamination, and then there are 2 transporters which are bringing the ammonia regularly. One is the alanine and another is the glutamine. Now, once the ammonia is here, now the liver is loaded with the ammonia. So much ammonia is there. This ammonia will be converted into its ultimate product, that is going to be urea. That is going to be step number 4. When the ammonia converts into urea, this is called as the urea cycle. And then this urea molecule will come in the blood. When the urea molecule comes in the blood, this is called as the blood urea nitrogen, which you have seen in the MCQs and in the lab reports written as BUN, blood urea nitrogen. And this blood urea nitrogen will reach all the way in the kidney and will be excreted out. So now we have understood that in the process of biosynthesis of urea, there are total 4 steps. Let's revise with me very quickly. First, you convert all the amino acid into which amino acid? Glutamate. And this process is called as what? Transamination. This will occur where? Hepatic or extrahepatic? Extrahepatic. Then this glutamate will go all the way to the liver. And what you are going to take out from the glutamate? Ammonia molecule in the presence of oxygen. So this is called as oxidative deamination. Then the step number 3 is going to be the transporters. We have 2 transporters of ammonia, alanine and glutamine. Who is going to bring the ammonia from the muscle? Alanine. From the brain? Glutamine. And now the liver is loaded with ammonia. And once there is so much of ammonia is there, it will convert into urea via the urea cycle. And then the urea will go all the way to the kidney and will be thrown out. So now we have got a basic idea that there are going to be 4 stages or the 4 steps: the transamination, the oxidative deamination, the transporters, and the urea cycle. Now we will discuss the details of each of them one by one. Starting with the first one, the transamination. I want some input from your side. When it comes to transamination, first tell me, it will occur where? Hepatic or extrahepatic? Transamination, yes, it is going to occur in extrahepatic tissue. Whenever we do any reaction, the agenda has to be very clear. The goal has to be very clear. When you want to do transamination, you want to convert the different amino acid into which type of amino acid? You want to convert all these amino acid into glutamate. This is the product. And what will the substrate? The different amino acids. The different amino acid. What will the name of enzyme? The name of enzyme is transaminase. And when we want to do the transaminases, we require a coenzyme, that is the active form of vitamin B6. The active form of vitamin B6, that is also called as the pyridoxal phosphate. In short, we will write pyridoxal phosphate as PLP. So this is also itself is a MCQ, that what is the coenzyme for transamination? It is PLP, pyridoxal phosphate. So the transamination is an easy process, that different amino acid you will convert into glutamate. You will convert alanine into glutamate, aspartate, glutamate, glycine to glutamate. But the problem is you can't remember all these reactions, right? We can't remember all of them. So we are going to see some examples of transamination. Means, for example, how the aspartate amino acid converts into glutamate. This is one example that we are going to discuss. See how it is going to happen. The product is fixed, that is glutamate. We have the different amino acid, maybe aspartate, maybe alanine. We will see some of them. So when the aspartate convert into glutamate, you require, for this transamination, you require alpha ketoglutarate. And with the help of enzyme transaminase and the vitamin B6, we are going to generate the glutamate. Along with the glutamate, we are going to generate one more byproduct, that is oxaloacetate. When we do the transamination of aspartate, we are going to generate the glutamate plus oxaloacetate. The name of the enzyme, because it's a transamination, so it's a transaminase. But if I ask you, what is the specific name of this enzyme? The specific name of this enzyme is serum glutamate oxaloacetate transaminase. Serum glutamate oxaloacetate transaminase, which you have also written as SGOT and seen in the many reports, right? SGOT. This SGOT is basically doing the transamination of the aspartate. So this SGOT, also called as aspartate transaminase, written as AST, aspartate transaminase, right? The full form is written below. Let's see one more example. If I want to convert, for example, alanine amino acid, I want to do the transamination and I want to convert into glutamate. The bisubstrate, again alpha ketoglutarate. The byproduct here we are going to get is pyruvate. So what will the name of enzyme of this transamination reaction? It will be like this: serum glutamate pyruvate transaminase. And because this SGPT is doing the transamination of alanine, so this is also called as alanine transaminase, which is written as ALT. And we will require vitamin B6. Obviously, we can't remember all the transamination reaction, so we'll just remember two transamination reaction: the aspartate, how it converts into glutamate, and how the alanine converts into glutamate. In the transamination reaction, the product is fixed, glutamate. What is the byproduct? That is the part to be remembered. So we are going to use a mnemonic for this, and we will be using this mnemonic multiple times in the different areas of biochemistry. So you will remember it automatically. So we have a weird mnemonic with the help of SOLP. What is the meaning of SOLP here is S stands for aspartate. Aspartate, when it converts into glutamate, the byproduct is oxaloacetate. When it comes to the alanine, L stands for the alanine. When you do the transamination, the byproduct you are going to get is pyruvate. Because the product is fixed. See, in these reaction, this area is fixed. The glutamate is fixed in all the transamination. In fact, the bisubstrate is also fixed, alpha ketoglutarate. So we don't have to remember them. We just have to remember the substrate and the byproduct. And we will remember with the help of the name of the enzyme, SOLP. So what is transamination? Transamination is basically we are converting the different amino acid into glutamate. And can you answer me why? Why to glutamate only? Why not to glycine? Why not to any other amino acid? Because glutamate has a property. It can easily donate which group? It easily donates the ammonia. And this is what we wanted, right? So there are three more amino acids which can also do the same. They can easily also donate the ammonia. Then for those amino acids, we don't have to do the transamination. They can directly go to the liver and can donate the ammonia. Right? What are the name of these amino acids? The name of these amino acid is TPL. It is not IPL, TPL. Threonine, proline and lysine. Threonine, proline and lysine does not undergo transamination. They does not undergo transamination. Instead, they directly go to the liver and donate their ammonia. So they ask this question that which amino acid does not undergo transamination? The mnemonic is TPL, not IPL, TPL, threonine, proline, lysine. So we have understood the transamination, and in the important part in the transamination was the two enzymes, the SGOT, SGPT. There is so much of clinical importance is there for SGOT, SGPT. Let's try to understand them. See, we have discussed the SGOT, SGPT you have seen in the liver diseases. What we can do is, liver disease we can divide into two categories. Here, let's understand. In the gastrology also you discuss about this. The liver diseases can be divided into two categories. Either it is because of alcoholic or it is non-alcoholic. It is seen that in the alcoholic liver disease, the SGOT is going to increase significantly more than the SGPT if there is alcoholic hepatitis. the SGOT will increase significantly more. Whereas the non-alcoholic liver disease, for example, infectious hepatitis, hepatitis B, in that case, the SGPT is going to increase significantly more than the SGOT. So if I make a ratio, here this ratio is going to be more than 2, SGOT SGPT ratio, and here in the non-alcoholic liver disease it will be less than 2. But we know that the alcoholic liver disease or the non-alcoholic liver disease, ultimately they can reach all the way up to cirrhosis. If the cirrhosis develops, whether it is alcoholic or non-alcoholic, because hepatitis B can also lead to cirrhosis, alcohol can lead to cirrhosis. If the cirrhosis develops, can you tell me that in cirrhosis patient which is more, SGOT increases more or SGPT increases more? In the cirrhosis patient, it is SGOT which is going to be more than SGPT. So whether it is alcoholic or non-alcoholic cirrhosis, SGOT will be more. But if there is hepatitis, then you have to see that which type, alcoholic or non-alcoholic. Alcoholic will have more of the SGOT increase. In the non-alcoholic, it is going to be the SGPT which will increase more. The same data is written here. When it comes to the ratio is more than 2, it's the alcoholic liver disease. It is less than 1 or less than 2, we can say it's non-alcoholic liver disease. SGOT is just not found in the liver cells. It's not just found in the liver cells. They are also found in the skeletal muscle. They are also found in the cardiac muscle. SGOT is also found in cardiac muscle. Means what? In the case of rhabdomyolysis means the muscle injury. Because the muscle contains a lot of SGOT, a lot of SGOT will come out. And significant increase in the SGOT will be there, and the ratio is going to be more than 5. In the case of MI, myocardial infarction, because the cardiomyocyte also contains the SGOT, so in the case of MI, there is going to be increase in the SGOT level. In cirrhosis, whether it is alcoholic or non-alcoholic, what is going to increase more? Yes, it is SGOT is going to increase more. So SGOT, we have understood that it is increasing in 4 conditions. Let's revise with me. Alcoholic liver disease. Second, it is going to be the rhabdomyolysis. Third is myocardial infarction. And fourth is any cause of cirrhosis. What are the other important areas near to this? The SGOT, SGPT, if it is in thousands, normally SGOT, SGPT is 30-40 international unit. But if the SGOT, SGPT is given in thousands, what can be the causes? There are only few causes, such as autoimmune hepatitis, ischemic hepatitis, drug-induced hepatitis or the drug toxicity. The most common drug that leads to hepatitis or the liver damage, that is acetaminophen. So if the SGOT, SGPT, it is in 1000, it can be because of autoimmune hepatitis, it can be seen in the ischemic hepatitis or drug toxicity. Even in sometime in the Wilson disease also we can have the SGOT, SGPT in thousands. So this is the basic idea behind the liver function test. In the when you interpretate the SGOT, SGPT, how to interpret the SGOT, SGPT, we got a basic idea. Now here itself I want to add some more point from the liver function test. We have understood the idea behind the SGPT and SGOT. There is one more thing that you have seen in the liver function test, that is alkaline phosphate. Alkaline phosphate values. See, alkaline phosphate will be increased if any time there is the biliary obstruction. Whenever there is a biliary obstruction, alkaline phosphate will increase. Actually, in biliary obstruction, there are 3 enzymes which are related to biliary obstruction. One is the alkaline phosphate, but alkaline phosphate is very non-specific. Can you tell me some more which is going to be increased? Yes, it is gamma glutamyl transferase, GGT. If the GGT is increased, it is again the biliary obstruction. If there is increase in 5' nucleotidase, that is again biliary obstruction, means the bile flow is not happening properly. SGOT, SGPT, they are related to the hepatocellular injury. Now, alkaline phosphate, I told you that it is going to increase in the biliary obstruction, but it is very non-specific. Actually, alkaline phosphate can be generated from different tissues. What all tissues which are related to alkaline phosphate? Can you tell me what all tissues are related to alkaline phosphate? Yes, one, it is the bone. If there is any problem in the bone, alkaline phosphate can increase. It is also coming from placenta. It is there in the liver. Alkaline phosphate is related to kidney disease also. and it is also related to intestine. So alkaline phosphate is actually related to 5 tissues. The mnemonic that we are going to use is BPL kit. BPL kit. Bone, placenta, liver, and intestine. BPL kit. Right? So let's summarize the data. Whenever they talk about the SGOT, SGPT, they are related to hepatocellular injury. If there is an alcoholic hepatocellular injury, SGOT will increase. If non-alcoholic, SGPT will increase. And if there is cirrhosis because of any cause, the SGOT will increase. Secondly, we have discussed that if there is a bile obstruction, maybe because of gallstone or maybe because of tumor, then there are 3 enzymes which can increase. One is alkaline phosphate, the second is going to be the GGT, and the third is the 5' nucleotidase. The GGT has been asked recently in the exam. They asked that which molecule or which enzyme is going to increase in a patient of biliary obstruction. It is going to be the GGT. So let's come back to our topic. We were doing the biosynthesis of urea. All the amino acid will, first step is all the amino acid will try to convert into glutamate, and that is called as transamination. And why you do that? Because the glutamate can easily donate the ammonia. Can you tell me the 3 amino acid which does not undergo transamination but they directly go to the liver and donates their ammonia? What is the mnemonic for that? TPL, threonine proline lysine. They can directly go. Now coming to the step number 2, that is the glutamate has reached to the liver, and in the liver we are going to take out the ammonia from the glutamate. But before I start that, I want to tell you one thumb rule. There are total 3 thumb rules that we are going to follow in the biochemistry, and once you will familiar with these thumb rules, the things will become very easy for you. So here comes the first thumb rule. In any reaction of biochemistry, if you see NAD NADH or FAD FADH, if you see these things NAD NADH or FAD FADH, the name of the enzyme is going to be dehydrogenase. This is the first thumb rule for us. The name of the enzyme is going to be dehydrogenase. NAD NADH FAD FADH. Let's see the step number 2, oxidative deamination. Can you tell me where it is going to occur? Extrahepatic or hepatic tissue? It is going to occur in the hepatic tissue. And I want the answer from your side now. That the substrate is going to be glutamate. Glutamate has reached in the liver. What you want to take out from the glutamate? What you want to take out in the oxidative deamination? What is the product? Ammonia. And in this reaction, NAD converts into NADH. Whenever NAD NADH comes, what is the name of enzyme? The name of the enzyme is glutamate dehydrogenase. NAD NADH is dehydrogenase. And because it's an oxidative process is going to occur in the mitochondria because oxygen is present in mitochondria. So I can say it will present the oxidative deamination, it is going to occur in the mitochondria. So the oxidative deamination is a very easy process that from the glutamate will take out the ammonia, and that is because the NAD NADH comes into the reaction. So the name of enzyme is dehydrogenase. Now the liver is having ammonia. The third step, there are some free ammonia molecules are there in our body which we have to bring to the liver, and that is the transport of the ammonia. So how we are going to transport the ammonia? First we will learn from skeletal muscle how we are going to bring the ammonia all the way to liver. So for example, here are the free ammonia molecules are there in the muscle. I want to bring them to the liver. For the energy purpose, what the muscle will do is the muscle uses the glucose molecules, and the glucose molecule undergoes glycolysis. The product is going to be the pyruvate that we will discuss in the glycolysis. In the carbohydrate part, the glucose converts into pyruvate and give the energy to the muscle. Now if there is free ammonia molecules are there and you want to bring it to the liver, the ammonia molecule can club with the pyruvate and can join and can make a molecule called as alanine. And now this alanine molecule will come in the blood, and via blood this alanine molecule will reach all the way to the liver. Once the alanine will reach there, this alanine will bifurcate back into the ammonia molecules and the pyruvate. And this is what we wanted. Basically we wanted the ammonia molecule to reach all the way from the muscle to liver, and we have done that. And with the help of which molecule? Alanine. So they asked this question, what is the transporter of ammonia from the muscle to liver? The answer is alanine. It's a very important MCQ. Although the task of transport is done, but now I want to add one more point here. We will understand that what is going to happen with the ammonia. We know that it is going to convert into urea, but what is going to happen with the pyruvate? This pyruvate can convert into glucose, and this process is called as the gluconeogenesis. And now this glucose molecule can come back in the blood and can go all the way to the muscle for the energy purpose. So this entire thing is called as the Cahill cycle. The agenda was not to understand the entire Cahill cycle as of now. Just remember one simple statement: who brings the ammonia from muscle? Very important MCQ. It is alanine. Let's understand how we are going to bring the ammonia from the brain and the other tissues. So for example, here is the ammonia molecules in the brain, and I want to bring it. So these ammonia molecules are going to club with the glutamate molecule, and they are going to make a molecule called as glutamine. This glutamine molecule can travel all the way to the liver, and once the glutamine will reach there, this glutamine can bifurcate into two. It can bifurcate back into the ammonia and the glutamate, and this is what we wanted. Actually, we want to bring the ammonia from the brain to the liver. And who has helped in this process? Glutamine. So they ask this: who is the transporter? The transporter is glutamine. To synthesize the glutamine, the name of the enzyme is glutamine synthase, and this is how the glutamine will reach there. Now once the glutamate reaches in the liver, it will bifurcate into the ammonia and glutamate. Ammonia, we understand that. What is going to happen with ammonia, we will understand that. But what is going to happen with this glutamate? This glutamate can undergo oxidative deamination, and it can also generate the ammonia. Oxidative deamination, just now we have discussed about the oxidative deamination. The oxidative deamination means you take out the ammonia. So this is how we transport the ammonia from the brain and the most of the tissues. The glutamine can also go all the way to the kidney also, sometimes, not the majority of the part, but some percent can go to the kidney, and in the kidney the glutamine can convert into glutamate plus ammonia. So actually the glutamine can donate the ammonia into two tissues. One is the liver and second is in the kidney. In the kidney it's going to be very fractional, but it is there. So sometimes they ask that the free ammonia can be generated into two tissues. What are the two tissues where the ammonia can be transported? It is, one is the liver and second sometime in the kidney also. So far what we have discussed, let's revise it. Very important. First of all, in the biosynthesis of urea, you have converted all the amino acid into which amino acid? Glutamate. This process is called as transamination. Now the glutamate will go all the way to the liver, and once the glutamate comes in the liver, you are going to take out the ammonia. That is called as oxidative deamination. Then we have some free ammonia molecules in different part. You are going to send that also. And how we are going to send that? With the help of the transporters. Who is the transporter of ammonia from the muscle? Alanine. From the brain, glutamine. And this is how the ammonia reaches in your liver. The liver is full of ammonia now. And then we will convert the ammonia into urea. Now, before I go to the step number 4, I want to learn you the second thumb rule. The first was very easy. Whenever there is NAD, NADH or FAD, FADH, that is dehydrogenase. The second thumb rule is to understand the difference between the enzyme synthase and synthetase. See, whenever we have two small molecules and they club together and they make a big molecule, either it is a synthase reaction or synthetase reaction. How to decide that? If two small molecules are joining and making a big molecule without any ATP, if the ATP is not used, no energy is used, then the name of enzyme is synthase. But if you are making a bigger molecule with the help of energy, then the name of enzyme is synthetase. So we have synthase and synthetase. Small molecule joining together, making a big molecule. We want to see the ATP is used or not used. If the ATP is not used, it is synthase. If the ATP is used, synthetase. So now, what we are going to do with the ammonia, which has came all the way in the liver. Just now I told you the ammonia was coming in the liver by these three methods. One was the oxidative deamination of the glutamate. Second was the transporter. One was coming from the muscle, that is the alanine. Alanine has came from the muscle, and the brain, the glutamine has came. So now we are having so much of ammonia. Now you will answer. The ammonia will be converted into what? Urea. And that process is called as what? Urea cycle. So now we will see the details of the urea cycle. Urea cycle, it is also called as the ornithine cycle. It is also called as the Krebs-Henselt cycle. Where it is going to occur? The organ is going to be the liver. But in the liver, in the hepatocytes, where this urea cycle will occur, in the cytoplasm or mitochondria. Actually, some reaction occurs in the mitochondria and some reaction occurs in the cytoplasm. There are total 5 reactions are there in the urea cycle. Total 5 reactions. Out of 5 reactions, the first 2 reactions, first and second, they will occur in the mitochondria, whereas the third, fourth, and the fifth reaction they will occur in the cytoplasm. In the urea cycle, the agenda is very clear that we are having a ammonia molecule and we want to convert that into urea. This is the agenda. There will be total 5 reactions. The first 2 reactions will occur in the mitochondria and the remaining 3 reactions will occur in the cytoplasm. So here is the representation of the hepatocyte. This double membrane structure, this is the mitochondria, and outside the mitochondria, this is the cytoplasm. There are 2 reactions which is going to occur in the mitochondria. So the first starting molecule is going to be the ammonia. This is the starting molecule. We will do 5 reactions. The step number 1, step number 2, these are 2 in the mitochondria. Then we'll do the step number 3, 4, and 5 in the cytoplasm. And what is the ultimate product you want from the urea cycle? What is the ultimate product you want? Urea, which we are going to generate. So because once you know the target, then the things becomes easy. So that is why in every pathway I tell you that what is the ultimate agenda. We are having ammonia, we want to convert into urea. So the first step, the ammonia molecule and the carbon dioxide molecule, one molecule each, are going to join together with the help of energy. With the help of ATPs, we are going to use 2 ATPs and we are going to make a big molecule called as carbamyl phosphate. Because we are making a big molecule with the help of energy, what do you say? What should be the name of enzyme? Synthase or synthetase? What should be the name of enzyme? Yes, it is going to be synthetase. So it is carbamyl phosphate synthetase 1. In short, we are going to write as CPS1. This CPS1 is a very lazy enzyme. And in the entire pathway, whoever is the most lazy enzyme, that is called as the rate limiting step. The CPS1 is the most lazy enzyme of the urea cycle. So this is called as the rate limiting step. It is so lazy that to activate the CPS1, we need an activator. And the activator of the CPS1, it is NAG. So NAG is going to activate the CPS1. NAG is the N-acetyl glutamate. It is going to be the activator of the CPS1. And because it is the most lazy enzyme, so this is the rate limiting step, CPS1. Now we have the carbamyl phosphate. Now, for the carbamyl phosphate, there is one molecule which is present here in the mitochondria with the name of ornithine. So the carbamyl phosphate now react with the ornithine. And how they are going to react with the ornithine is, from the carbamyl phosphate, we will take the carbamyl group and we are going to transfer the carbamyl group on the ornithine. If you transfer the carbamyl group, that is called as transcarbamylase. And because we are transferring on the ornithine, so the name of enzyme will be ornithine transcarbamylase. So I am writing the short form. The full form is written below. The name of the enzyme in the second step is ornithine transcarbamylase. Now, once you transfer the carbamyl group on the ornithine, the molecule that you are going to generate, it is called as citrulline. The citrulline will come out now from the mitochondria to the cytoplasm. When the citrulline comes out, in the step number 3, there is a foreign particle which is going to join the urea cycle. In the latter half, the name of this molecule is aspartate. So now the citrulline and aspartate will club together and will make a big molecule. The big molecule they are going to generate is arginosuccinate. And to make this big molecule, we require ATP also. And whenever 2 small molecules joins together and makes a big molecule with the help of energy, what will the name of enzyme? Synthase or synthetase? Yes, it is synthetase. So the name of enzyme is arginosuccinate synthetase. In this process, we require ATP, adenosine triphosphate. It converts into AMP. The triphosphate will convert into monophosphate. Triphosphate converts into monophosphate. Means how many phosphate are utilized here? 2 phosphate. This arginosuccinate is a very big molecule. This arginosuccinate will split into 2 small molecules. The 2 small molecules it is going to split, arginine and fumarate. Arginine and fumarate. When the arginosuccinate is split into 2 small molecules, arginine and fumarate, It's a lysis reaction. So the name of enzyme is arginosuccinate lyase. Or we can simply write as arginosuccinase. Arginosuccinate lyase or arginosuccinase. Then this arginine is going to further split into two small molecules. It's going to further undergo lysis reaction but with the help of water. And now it is going to split into two. One is your dream molecule that we were willing for last 40 minutes or so, that is urea, and one more molecule that will be generated, ornithine. So arginine is going to split into these two molecules with the help of water. What do you think? What should I say? Is it lysis or hydrolysis? What is the better name? Hydrolysis. So the name of enzyme is arginine hydrolase. This arginine hydrolase is also called as argininase. Argininase. So these are the 5 reactions. And we have got our dream molecule, that is urea. This urea can easily come in the blood, and when the urea comes in the blood, this is called as what? Blood urea nitrogen burn. And this burn will go all the way to the kidney and will be thrown out in the urine. But what is going to happen with this ornithine molecule? This ornithine will be reutilized. It will go inside the mitochondria and it will be reutilized. To transfer the ornithine, we require the transporter, that is ornithine transporter 1. Ornithine transporter 1. It is going to transport the ornithine from the cytoplasm to the mitochondria. Basically, the technical name of this ornithine transporter is the solute channel, SLC. SLC means, SL means solute, C means channel. Solute channel means it is transferring the solute, means the particle of ornithine. Solute channel 25A15. 25A15, that is basically transferring the ornithine from the cytoplasm to the mitochondria. And that is why the urea cycle is also called as ornithine cycle. You can see that the ornithine is getting reused. It is getting reused, right? So this is all about the urea cycle, and this is how we make the urea. These are the 5 steps. But you know that biochemistry, one of the technical problem with this subject is that there are so many reactions, there are so many pathways. How we are going to remember all the pathways? See, the idea is, no, in these competitive exam, be it NICT or the NEET PG or the FMG exam, no one is going to ask you to write down the urea cycle or the Krebs cycle, right? They ask some particular questions, some particular points. So after every pathway, I will tell you that what are the important points to be remembered, and that is the important idea, right? So when you are revising the notes, don't write this urea cycle for 10 times, 20 times and cram the data. Anyway, you are not going to remember that, but the important points has to be remembered, which we are going to write after every pathway. We will remember the points to remember. What you can do is, when you will read the notes multiple times, 6 times, 7 times, when you revise the notes multiple times, automatically the things will settle down. Instead of putting the energy in the first go, writing the urea cycle for 10 times, and then, because if you put so much of energy today, and your NEET PG exam is, say for example, after 8 months or 7 months, in this duration, if you don't reopen it again, then it is going to be wiped out. Towards the end, when you will open the notes again for revision, then you will not be recalling that what were the intermediates of urea cycle. So instead of putting the energy today, what you do is, you just read these pathways, and then points to be remembered has to be given more attention. So when it comes to the points to remember, when it comes to the energetics of urea cycle, actually we have invested the energy 2 times, in the first reaction and in the third reaction. In the first reaction, we have used 2 ATPs, which converts into 2 ADPs. So I can say, in the first reaction, how many ATPs has been used? 2 ATPs. And how many phosphate has been used? Triphosphate is converting into diphosphate, means 1 phosphate is missing, and this has happened how many times? 2 times. So 2 phosphate used. In the reaction number 3, the triphosphate has converted into monophosphate. How many ATP used? 1. But how many phosphate used? 2. So if they ask total ATP used in the urea cycle, 3 ATPs, but total phosphate used, 4 phosphates. What is the rate limiting enzyme? The rate limiting enzyme is the most lazy enzyme. Is it CPS1 or CPS2? CPS1. Who is the activator of CPS1? NAG. Okay, total how many reactions were there in urea cycle? 5. Means there are 5 enzymes. Sometimes they ask a question, what is the 6th enzyme of urea cycle? Actually, this NAG is considered as the sixth enzyme of urea cycle. NAG is considered as the sixth enzyme of urea cycle. Then, because two reactions are occurring in mitochondria, three are occurring in cytoplasm, and whenever there is a combination of three, it's a potential all except quotient. So they ask all of the following are cytoplasmic enzyme of urea cycle except. So we should know which is cytoplasmic enzyme and which is mitochondrial enzyme. The cytoplasmic enzyme, look at the names of the cytoplasmic enzyme. Look at the name. Arginosuccinate synthetase, arginosuccinate lyase, and arginine hydrolase. They are starting with the alphabet A. So we can remember like this. Cytoplasmic enzymes of urea cycle, they are going to start with letter A. Cytoplasmic enzymes of urea cycle starts with the alphabet A. This is the urea that we have synthesized. This is the formula of urea, NH2CONH2. If you're listening to the words very carefully, I told you that in the first reaction, how many ammonia molecules and how many carbon dioxide you started with? You started with one molecule only. So in the urea, the formula is like this, NH2CONH2. This first NH2 group, it is coming from the ammonia particle, from the ammonia. This CO, it is coming from the carbon dioxide. But from where this second nitrogen is coming, from where this second amino group is coming in the urea molecule? Can you tell me one particle which was joining the urea cycle in the latter half, the foreign particle? What was the name of that? Yes, aspartate. So very important MCQ that in the urea cycle there are two nitrogens. Who donates these two nitrogen? One is from the ammonia and second is coming from what? It is coming from aspartate. So ammonium and aspartate, they are giving the two nitrogens of the urea molecule. So these are the important points, the physiological points to be remembered from the urea cycle. Then there are some disorders which are related with the urea cycle. If the urea cycle is not working properly, how the patient will present, what will be the manifestations of them? This is what we have discussed in the urea cycle. The same thing is written here. Actually, the ammonia ultimately converts into urea and there are total 5 reactions. The first 2 reactions are occurring inside the mitochondria and the remaining 3 reactions are occurring in the cytoplasm, and ultimately we will making the urea and ornithine. This ornithine will come back and with the help of ornithine transporter 1 it will come inside and it will be reutilized. This is what we have discussed, right? Now I ask a simple question to you. If by any chance, if the urea cycle is not working, means the urea will not be generated. Actually, the ammonia will not be converted into urea. So what is going to happen with the ammonia levels? If by any chance, if the urea cycle is not working, means the ammonia is not converting into urea, the ammonia is going to increase. And what is going to happen with the urea levels? Decrease. The urea in the blood is called as the BUN. The BUN is going to decrease. So, for example, what was the name of the first enzyme in the here? CPS1. What was the name of the second enzyme? Ornithine transcarbamylase. What was the name of the third enzyme? That was arginosuccinate synthetase or synthase. It is arginosuccinate synthetase because we have used the energy. Then the arginosuccinate lyase and then argininase. Right? These were the enzymes that we have used in the urea cycle. If by any chance, if any of the enzyme is not there, the ammonia will start accumulating in the body and that is called as hyperammonemia. So if there is deficiency of CPS1, if there is deficiency of CPS1, then this reaction will not occur and ultimately the ammonia will increase. That is called as hyperammonemia type 1. If there is deficiency of ornithine transcarbamylase, means this reaction will not occur and ultimately carbamyl phosphate will accumulate and ultimately ammonia will accumulate. That is hyperammonemia. And whenever any problem is there, the urea will not be synthesized, the urea will decrease, means the blood urea nitrogen will decrease. Blood urea nitrogen will decrease. In fact, if for example, this ornithine transporter is not working, let's say, this is not working, the ornithine will not go inside. If the ornithine will not go inside, again the urea cycle can't occur and hyperammonemia will occur. For that matter, if there is deficiency of this enzyme, then this reaction will not occur and ultimately the citrulline will accumulate and ultimately the ammonia will accumulate. So this disease, if the arginosuccinate synthetase is not working, the citrulline is going to accumulate and this is also called as citrullinemia. In this disease also we are going to have the hyperammonemia, but this disease is called as citrullinemia. If for example, if this enzyme is not there, arginosuccinate lyase is not there, this is called as arginosuccinate urea or arginosuccinatemia. Right, so basic idea is very simple that if any enzyme of urea cycle is not there, the ammonia is going to accumulate and the urea is going to decrease. But for example, they have given you a question that there is a patient who is having more ammonia, less urea. Where is the problem? Is it hyperammonia 1? Is it hyperammonia 2? Is it the problem of the transporter or something else? So how we are going to identify? Let's understand them one by one. First, the hyperammonia type 1 and type 2. Hyperammonia type 1 is due to deficiency of CPS1, type 2 is due to deficiency of ornithine transcarbamylase. In both the condition, the ammonia levels will be more and the blood urea nitrogen will be less. In both the condition, ammonia is more and the urea nitrogen is less. So if we are confused between hyperammonia type 1 and type 2, how we are going to identify? Hyperammonia type 2, it is associated with a special finding that we are going to have increase in the orotic acid, means there is going to be orotic acid urea. So in the MCQ, if they say there is a hyperammonia with orotic acid urea, or there is one more associated finding that there is going to be increase in the uracil in these patients of hyperammonia type 2, then you can easily identify that, okay, this is going to be the hyperammonia type 2. Let me explain you it here. Hyperammonia type 1, so you understand the problem in the first enzyme is the type 1. But for example, if patient is having the problem in the OTC, if there is deficiency of OTC, this reaction will not happen. The carboxyl phosphate will increase. The ammonia is also going to increase, but carboxyl phosphate will also increase. Because the carboxyl phosphate will increase, it can trigger an alternate pathway and it can convert into orotic acid. So that is why in the hyperammonia type 2, there is more carboxyl phosphate, so there is going to be more orotic acid. And the orotic acid can convert into uracil. So in a patient of hyperammonia, they are talking about uracil, or they are talking about orotic acid urea, they are talking about the type 2. Whereas in the type 1, the problem is in the CPS1. And in that case, the orotic acid will not increase. If this reaction is not happening, the orotic acid will not increase. So this is how we differentiate the type 1 from type 2. Then, for example, let's see here. We know that the ornithine goes inside via the ornithine transporter, that is the solute channel 25A15. With the help of this, the ornithine goes inside. Now, by chance, for example, this channel is not working. What is going to happen? If this channel is not working, the ornithine will not go inside. If the ornithine will not go inside, this reaction will not occur. If the ornithine is not there, this reaction will not occur. And ultimately, the urea cycle will not run, so the ammonia is going to accumulate. Ammonia is going to increase. Plus, there is few more things are going to happen. This ammonia, it is going to convert into carboxyl phosphate. Some molecules can convert into carboxyl phosphate. There is no problem. But now the ornithine is not there. So this carboxyl phosphate, because this enzyme was there, the transcarbamylase, it is going to convert into a special molecule. Normally it should convert into citrulline, but now because the ornithine is not there, so it is going to convert into a special molecule, that is homocitrulline. So in these patients, we are going to have increase in the homocitrulline also. There is increase in the ammonia, there is increase in the citrulline, homocitrulline. Plus, because the ornithine was not went inside, means the ornithine has not been consumed, so the ornithine is also going to increase. So if the ornithine transporter is not working, look here, if this transporter is not working, there are three things which are going to increase: the ammonia, the homocitrulline, and the ornithine. Hyperammonia, hyperornithinemia, and hyperhomocitrullinemia. This is called as triple S syndrome. So there is going to be, in the triple S syndrome, there is a defect in the ornithine transporter 1. There will be increase in the ammonia, decrease in the urea, plus there is going to be hyperornithinemia, plus homocitrullinemia. And that is why this disease is also called as triple S syndrome: hyperammonia, hyperornithinemia, and homocitrullinemia. Okay, we understood that. Let's keep it very simple. How we are going to solve the MCQ, how we are going to crack, what will be the hints for us? In any MCQ that talk about increase amount of ammonia and decrease amount of urea, you should think that there is some problem in the urea cycle. Then you have to see the associated finding. For example, if they say there is increase in the orotic acid, or they say increase in the uracil, if they are saying like this, they are talking about the hyperammonia type 2. In fact, let me add you one interesting point here. Increased amount of orotic acid can lead to macrocytic anemia also. can lead to macrocytic anemia. So if they say increase in the ammonia, decrease in the urea with macrocytic anemia, then also they are talking about the hyperammonemia type 2, because orotic acid can lead to the macrocytic anemia, hyperammonemia type 2. These are the hints. From here they can make a difficult question also. Let's say they make a question like this, that there is a patient, he's having macrocytic anemia, his B12 level and B9 level is normal. What can be the possible cause? The answer is going to be the orotic acid urea. So let me write you a box here, that is just extra information. When it comes to the causes of macrocytic anemia, see, macrocytic means the patient is having the MCV is increased. It can be because of vitamin B12 deficiency, vitamin B9 deficiency, and orotic acid urea. These are the various causes of macrocytic anemia. So they can ask you the question like this from the hematology also, that there is a patient, he's having macrocytic anemia, but his B12 and B9 level is normal. What can be the possible cause? The third possible cause that can lead to macrocytic anemia is orotic acid urea. In a patient of orotic acid urea, also we have the macrocytic anemia. So this is all about the hint for the hyperammonemia type 2. Hyperammonemia type 2. For the triple S syndrome, the hints are very easy. For the triple S syndrome, if any MCQ, they say that there is increase in the ammonia, plus there is increase in the ornithine, plus there is increase in homocitrulline, then they are talking about the triple S syndrome. Means there is a problem in the ornithine transport. One more hint that can be there is, if the ornithine is more, you know that the ornithine, sometime if it is much more, it is very toxic to the retina. Increased amount of ornithine can lead to damage to retina, choroid, it can damage there. And this damage is going to occur in a very peculiar manner, because if the ornithine is more, we are going to have the damage in the retina in a very peculiar manner. It looks like this. This is called as the gyrate atrophy. Gyrate atrophy. So gyrate atrophy is related to increased amount of ornithine. Then let's understand some more problems. For example, if they say, now you will answer this question, that there is a patient, he is having increase in the ammonia. The moment there is increase in the ammonia means there is a problem in the urea cycle. Plus there is increase in the citrulline. Increase in the citrulline. They ask, where is the problem? Where is the problem? Is it the problem of CPS1? Is it the problem of OTC? Is it the problem of arginosuccinate synthetase? So you will answer this question. Where is the problem? If the citrulline is more, ammonia is more as well as the citrulline is more. See here, for example, here is the ammonia, it converts into carbamoyl phosphate, then it converts into citrulline, then it converts into arginosuccinate synthase with the arginosuccinate with the help of enzyme synthetase. If there is deficiency of this enzyme, this reaction will not occur. Citrulline will increase, this will increase, this will increase. So if there is increase in the ammonia with increase in the citrulline, where is the problem? Arginosuccinate synthetase. Right? So if there is increase in the ammonia, increase in the citrulline, the problem is deficiency of arginosuccinate synthetase. In the same way, if there is increase in the ammonia with increase in the arginosuccinate, if the arginosuccinate is more, then where is the problem? If the arginosuccinate is also more, then the problem is the arginosuccinate split is not happening. If I again show you the same thing here in this diagram, see here, normally the ammonia converts here, citrulline to arginosuccinate and arginosuccinate to arginine. Now I am saying that if this enzyme is not there, there is deficiency of this enzyme, the reaction will not happen, arginosuccinate will increase, this will increase, this will increase, and this will increase. So if there is increase in the ammonia with increase in the arginosuccinate, means the problem is in arginosuccinate lyase. There is going to be deficiency of arginosuccinate lyase. And this increased amount of arginosuccinate, it is going to come in the urine, as well as it is going to damage the hairs also. So the patient's hairs will get damaged, they will easily break. So if they say, the hint will be hyperammonia with some problem in the hairs. Some problem in the hairs. There is only one disease with hyperammonia with hair problem. It is going to be the deficiency of arginosuccinate lyase, and this is called as the arginosuccinate urea. The disease is called as arginosuccinate urea. I know that it might look little difficult because we have discussed total 5 diseases related to the urea cycle, but all the 5 are important to be remembered for your exam, and you should be fast enough to make the diagnosis in the exam, that it should not take much than 30 seconds to make the diagnosis that what is the disease. So just to summarize this entire data, I can say like this: whenever there is increase in the ammonia, there are 5 diagnoses that should come in your mind. What are the 5 diagnoses? Is it hyperammonia type 1? Is it hyperammonia type 2? Triple S syndrome? Hypercitrullinemia? Or arginosuccinate urea? These are the 5 diagnoses that should come in your mind whenever there is increase in the ammonia. Then how we are going to make the diagnosis? The type 1, there is no associated finding. In the type 2, we are going to have increase in the orotic acid and increase in the uracil. If you see these words, they are talking about the type 2. In the triple S syndrome, they will say increase in the homocitrulline or increase in the ornithine. And you know that ornithine leads to which problem in the retina? Gyrate atrophy. If they talk about gyrate atrophy, they are talking about the triple S syndrome. In this, there is going to be increase in the ammonia with increase in the citrulline. And here, there is going to be increase in the ammonia with hair problem, hair defects. That is arginosuccinate urea. So this is going to be the table of the hints for us. It may take little time, but if you know the steps, and if you know that if there is a problem here, ultimately the ammonia is going to increase, you just have to remember the associated finding. It is important to invest the time in this particular table, because there are so many MCQs you are going to encounter which can be easily solved from this particular part. Now let's have one more clinical correlation here. See, what is happening is, right now, whatever protein you are eating in your diet, this protein is getting absorbed in the intestine, and ultimately it is getting metabolized. And one of the product that you are getting from the protein metabolism, it is ammonia. And this ammonia is easily getting converted into urea with the help of the liver. In our liver, there is one more place from where the ammonia is coming. It is from the commensal bacteria, the good bacteria which is living in your intestine. They also generate ammonia. So actually there are 2 sources of ammonia we have. The ammonia, the protein which you are eating in the diet, that is generating ammonia, and your intestinal bacteria is also generating the ammonia. This ammonia will be easily converted into urea with the help of the urea cycle in the liver. But for example, there is a patient, his name is Dharmendra. Dharmendra is chronic alcoholic, and because he is having chronic alcoholism, so he has developed cirrhosis of the liver. Means the liver has been damaged. If the liver is not working, means the ammonia will not convert into urea. If there is a cirrhosis, if there is cirrhosis, the liver will not work, and the ammonia will not be able to convert into urea, because liver is the only factory where you can convert the ammonia into urea. In cirrhosis, it will not happen. So there is going to increase in the ammonia. If there is increase in the ammonia, ammonia will come in the blood. And this increased amount of ammonia, because it will come in the blood, it is going to reach in the brain. If the ammonia molecules will reach in the brain more and more, because ammonia is a hyperosmotic molecule, it is going to attract the water. Slowly, this patient is going to develop the cerebral edema. So what is happening is, you can see that the problem was in the liver. It was cirrhosis. It was a hepatic problem. But ultimately, who is suffering? Brain. Because of liver problem, if the brain is suffering, this disease is called as hepatic encephalopathy. This disease is called as hepatic encephalopathy. How they are going to present? So for example, how this Dharmendra is going to present to us. The ammonia will increase more and more, and now the changes will happen in the brain. One of the first thing that he is going to notice is the altered sleep pattern. So one of the earliest thing that he is going to have is the altered sleep pattern. He's going to have change in his handwriting, that is dysgraphia. Now, altered sleep pattern means he will be sleeping more in the daytime, and nighttime he's not sleeping. Then slowly the drowsiness will increase more and more. After some time, he will be so drowsy that is most of the time it looks as if he is sleeping. But if you give him small stimulus, he will wake up. He is drowsy, but he will get up. But after some time, he will become so drowsy that with small stimulus, he will not get up. You have to give some painful stimulus, then he will get up. And after some time, the brain damage is so much that he will go into coma. So the patient will have altered sleep pattern, will have dysgraphia, will have the somnolence, the daytime somnolence. There can be seizure, decrease in the muscle tone, slurred speech, things can be there. Now he will come to you. Dharmendra will come to you. His family members will bring that he's not sleeping properly. He's sleeping more in the daytime. Night time he's not sleeping, and this is that the problems are happening. He's a chronic alcoholic. You know he's having a history of cirrhosis. It is hepatic NCAF. If you do the objective examination, you are going to have a special type of tremors will be seen. You will tell the Dharmendra to do like this and pull the wrist like this. He will not be able to hold it. His wrist will flap like this again and again. This is called as the flapping tremor. So in hepatic encephalopathy, we can have the flapping tremor. If you do the investigation, now you will answer these all questions. See, what is happening in the Dharmendra is the ammonia is not able to convert into urea. So what is going to happen with the urea level? Decrease. The urea will decrease. The ammonia will be more. If you do the investigation, the ammonia is more, the blood urea nitrogen is less. And because he has developed cirrhosis, you know that in cirrhosis, what is going to happen with SGOT, SGPT? What is going to increase more? SGOT or PT? Yes, SGOT will increase more than the SGPT. You are thinking that, yes, it is going towards hepatic NCAF. Even you can do EEG also. In the EEG, you will have the triphasic waves, triphasic or biphasic waves. I will not go into detail of that you will discuss in the gastrology part, the triphasic, biphasic waves. For the diagnosis, we use a classification that is called as the West-Heaven classification. And now it is time to treat. You need to understand the problem and you will tell the treatment. See, the problem in Dharmendra is happening is because of ammonia. You want to decrease the ammonia levels. And from where the ammonia is coming actually in Dharmendra's body, from where the ammonia is coming, it is coming from the protein in the diet and from the intestinal bacteria. So you will tell this patient to eat more protein or less protein? Less protein. And how to kill the intestinal bacteria? You will give the antibiotic. Or you can give a laxative so that his stool frequency will increase and the gut flora will be washed out. So I can say that for the treatment, first thing, we are going to decrease the protein in the diet. The second thing, we'll give the antibiotic to kill the gut bacteria such as rifaximin. We'll give the laxative such as lactulose that is going to increase that is going to clear the intestinal microflora. Then we can also give phenyl butyrate. This is an important MCQ. What the phenyl butyrate will do is, this patient is having more ammonia. Phenyl butyrate is an ammonia scavenger. It is going to bind the free ammonia. It's an ammonia scavenger. So this is how we are going to treat the Dharmendra, the hepatic encephalopathy. Right. So there are total 6 diseases we have discussed. 5 diseases we have discussed with the urea cycle enzyme deficiency. And the last disease we have discussed about the hepatic NCAF, right? So this is all about the biosynthesis of urea. Let's summarize this chapter. Here is the summary and some questions are written for you. You can take a pause here and you can write down the answer and then you can restart it. So let's see the answers. When it comes to the coenzyme of transamination, what is the coenzyme of transamination? Yes, all the amino acid, it is converted into which amino acid? Glutamate. What is the coenzyme for transamination? Vitamin B6. Vitamin B6. When it comes to the transporter of ammonia from brain to liver, who is the transporter? Glutamate or glutamine? Yes, it is glutamine. And from skeletal muscle, who is the transporter? Alanine. These are the two very important MCQs. In the myocardial infarction, there are many markers. We will discuss them in the chapter of enzyme, but as of now, SGOT or SGPT, which is a marker of MI. Out of these two, SGOT. Urea cycle occurs in the liver. In the liver, first two reactions in the mitochondria, remaining three reactions in the cytoplasm. The cytoplasmic enzymes, which are the cytoplasmic enzymes of urea cycle? Among the given option, which are the cytoplasmic, starts with the letter A. So whichever option is starting with the letter A, these will be the answer. Arginine hydrolase, arginosuccinate lyase. NAG is an activator. NAG is an activator of which enzyme? CPS1 or OTC? CPS1. Nitrogen molecule in the urea. How many nitrogens are there in the urea? If you remember, what was the formula of urea? NH2CO NH2. So who gives these nitrogen? One is coming from ammonia and second is coming from aspartate. How many ATPs are required in urea cycle? 3 or 4? 3. How many phosphate is required? 4. Hyperammonemia with orotic acid urea. Orotic acid. The moment orotic acid with hyperammonemia, it is going to be which disease? Where is the problem? Hyperammonemia type 2. Hyperammonemia type 2. Flapping tremors. It is seen in hepatic encephalopathy. Actually, flapping tremor is seen in few conditions. So let's write down them. This has been already asked. It can be seen in hepatic encephalopathy. It can be seen in uremic encephalopathy. It can be seen in CO2 narcosis, means hypercarbia. There are 3 amino acid which does not undergo transamination. What was the mnemonic? IPL or TPL? It is TPL. Threonine, proline and lysine. They does not undergo transamination because they can directly donate the ammonia. When it comes to the urea cycle disorder, we have discussed the 5 diseases. If you remember, urea cycle disorder, we have discussed hyperammonemia type 1, type 2. Urea cycle disorders, they are autosomal recessive except hyperammonemia type 2. Hyperammonemia type 2, it is X-linked. Hyperammonemia type 2, it was due to deficiency of CPS1 or OTC. It is deficiency of OTC. I know it was a little lengthy chapter, but I will just pinpoint which is the most important area from majority of the MCQ will come. See, one is these two questions. What is the name of the transporter? Must remember. And second, this particular one. This table will give you the idea to solve many MCQs about the urea cycle disorder, and among them, all the diseases are going to be the autosomal recessive except hyperammonemia type 2, which is X-linked recessive. And in fact, that is the most common among all the urea cycle disorder. You can write here also in the notes. Among all the urea cycle disorder, this is the most common, hyperammonemia type 2. It is X-linked. It's most common, right? Thank you.